Hello everyone and welcome to today's Lightning Lunch session. We are joined by Radha and Linda and they will be talking about IR spectroscopy, there we go, and also a quick overview of the course. Now, you may be joining us on demand, and if you are joining us on demand, then we hope you do enjoy this session. There's plenty more sessions during these Lightning Lunch series that you can watch either on YouTube or on our on-demand platform. There will be a little bit of time for questions today, but if you do have any questions, you will be able to ask those at a later date. You can either contact us at outreach@ljmu or courses@ljmu. If you are watching live right now, then we can't see and we can't hear you, so just sit back and relax. There is that Q&A button that you can press if you do want to ask a question to us all today. We're aiming for the session to be under half an hour, so hopefully we'll be quite snappy and that you'll get to take away loads of information about the course as well as relevant topics. So I'm going to stop sharing my screen and I'm going to pass it over.
Hello, everyone. My name is Raed Al-Kassas. I'm the program leader of the Pharmaceutical Science. And today I'm going to talk about the Pharmaceutical and Cosmetic Science program. So the schedule for today I'm going to introduce the program and I will talk about the structure of the program and the topics and modules that the program covers. Then my lecture will be followed by a lecture on infrared spectroscopy. This is one of the topics that we teach in the program, and the lecture will be delivered by Dr. Linda Seaton. So Pharmaceutical and Cosmetic Science program is a 3-year full-time degree program. The program also includes optional industrial placement year, and foundation year is also available in the program. The program is supported by professionally relevant curriculum that is research informed, and it aims to develop proficient scientists that are ready for employment with transferable skills. So to talk about the structure of the program is designed around the 3 key themes. The first theme is chemistry and analysis, and this theme includes studying the properties of the pharmaceutically and cosmetically relevant molecules. The second theme is formulation and production, and this theme involves studying how to design these molecules into a suitable formulation. This will include either pharmaceutical formulation or a drug delivery system or cosmetic formulation. The third theme is physiology and biological activity, and this theme involves studying the interaction of these molecules with the human body. The structure of the course, this slide shows the structure of our program. As I mentioned earlier, our program consists of 3 years. Each year consists of 2 semesters, first semester and second semester. Every semester comprises of 3 modules. So in the first year, which is level 4, we teach physical properties, or the modules are physical properties, organic chemistry module, and principles of human biology and disease. In the second semester, the modules are formulation science, analytical chemistry, biologically active molecules. In year 2 or level 5, the modules are pharmaceutical formulation, principles of pharmacology, and sterile pharmaceutical products. That's for the first semester. Whereas in the second semester, the modules are pharmaceutical analysis, principles of cosmetic products, and synthetic and natural products. Now, the third year is a little bit different. So in the third year, 2 modules are taught in every semester. So in the first semester, the modules are advanced therapies and delivery systems. And the second module is advanced analytical techniques. Whereas in semester 2, industrial product development module and safety assessment of pharmaceutical and cosmetics, But in the third year, we introduce the final year projects. So the students carry out a final year project under the supervision of one of the lecturers. And these are projects that run over the whole year. And the students work in the lab to generate some data, analyze these data and discuss them, and then write a dissertation and also present the outcomes of the research by oral presentation or as oral presentation. So the program involves lectures, practicals, workshops, and tutorials. And we teach in classrooms. Also, some of the lectures are delivered online, and laboratory-based sessions as well. The program also includes laboratory-based sessions. This slide shows a typical week in the first year. And as you can see, the timetable is relaxed, so it's not too busy. And this is to give students enough time to work on their assignments and to conduct literature search by internet, and also to visit the library and to socialize as well. What are the career prospects? So pharmaceutical and cosmetic industries are knowledge-driven. They require a highly skilled workforce. We prepare our students and we give them the experience and the qualification to be able to work in this industry or in the pharmaceutical and cosmetic industries. There are many options for the graduates or for our graduates. So the career options are, they could work in research and development department in any of the pharmaceutical or cosmetic industry. They could work also in a production department or a quality control department or a regulation and licensing department or a clinical trials department. So as I mentioned, we provide the students the skills, the qualifications, and also knowledge to be able to work in industry and to perform their job properly. 90% of our graduates secure employments or further study 6 months after graduation. So in summary, the benefits of joining our program are study and industry relevant and research informed curriculum. Also, the students will gain hands-on experience of a range of laboratory-based techniques, become a proficient scientist, develop key transferable skills, and increase your employability. Finally, thank you, and I hope you enjoyed or have enjoyed the session. Thank you.
Okay, so I'm going to now give you some insight into some of the things that you might study on this course. It's being introduced in the A-level chemistry syllabus, so it's something that you need to know about, and it's deepened and embedded if you were to join this degree course in pharmaceutical and cosmetic science, but also if you did a chemistry degree course, pharmacy, or probably a forensic science course as well. So this falls into the branch of organic chemistry, which is the study of molecules containing carbon. So for example, we've got here methane. Carbon always has 4 bonds. And organic chemistry is the study of structure, properties, composition, and the reactions and synthesis of all organic compounds. So this is really important for things like pharmaceutical materials. So your asthma medication is an organic compound. Also for things like agricultural materials, so fertilizers and insecticides so that we can grow our food. And for industries like manufacturing plastics that we need for everything from your laptop to your contact lenses. So organic chemistry is really important for an awful lot of things that we use. Now, organic molecules have functional groups. And this is a specific collection of atoms within the larger molecules. And they have characteristic properties and behave in a predictable manner and undergo certain reactions. For pharmaceutical materials, it's the functional groups on the molecule that gives it the therapeutic effect, so that makes it interact with the cells in your body and cure your illness or have the effect that we need it to have. So, for example, you will have met in your GCSE studies alkenes which have a double bond between carbons. That's a functional group. Here's an alcohol. This has an OH group. So this is in the hand sanitizer that we're all using. Carboxylic acids have the OH group and also this carbonyl group, carbon double bonded to an oxygen. An aldehyde has the carbonyl and the carbons joined to a hydrogen. And ketones have the carbon joined to another group containing a carbon. So these are examples of functional groups, and we need to be able to identify these, and these can also help us to identify molecules. And one way that we can do that is using infrared spectroscopy. So as the name suggests, it uses infrared light, which is part of the electromagnetic spectrum. And just like ultraviolet light interacts with the cells in your body, so we get sunburnt in this bright sunshine, infrared interacts with molecules. And when infrared energy is interacting with a molecule, it causes the bonds, the chemical bonds, to vibrate. So we can have stretches, as we can see here. This is methane again, and two of the hydrogens are lengthening their bond at the same time, or here one's lengthening while one is going back to its original place. Or the bond angle can change so that the bond angle bends. These vibrations happen at specific frequencies, so specific energies of the infrared region. This is the instrument, the spectrometer, which can measure those vibrations, and it measures how much light is absorbed by the molecule to make that vibration happen. And it measures the exact frequencies that that absorption occurs at. So this allows us to identify which bonds are present in the sample that we're examining. And it's used to then produce a spectrum. So this is a graph. We have absorbance here on this axis, and we have the wavelength of the light on this axis. It's actually 1 over the wavelength. So the units are reciprocal centimeters, 1 over the centimeters, and we call them wave numbers. And that's a particular term that we have just in infrared spectroscopy. So where absorbance is very low or close to 0, then the graph is near the top of this axis. And when the molecule absorbs infrared light and a vibration happens, then we see a peak in the graph. And that happens at a particular frequency. And we can see that for this sample, we've got lots of vibrations. And that's typical because we've got lots of different bonds in the molecule, and each has their own vibrational frequency. So each bond will have its own peak in the graph. And that means that we can identify different types of bonds in the sample. And that can help us to know what reactions that sample will undergo, or even exactly what sample it is. This region we call the fingerprint region. And this is vibrations that happen in the carbon and hydrogen bonds and the carbon-carbon bonds. So they're present in nearly all molecules. But the exact pattern acts like a fingerprint, and that will be unique to that particular molecule. So here's an example of how we can identify functional groups. Here's an OH group. We get a very broad peak on the left of the spectrum. For a carbonyl group, we get a very sharp peak in the center of the spectrum. So that's a double bonded carbon to oxygen. Here we've got a single bonded carbon to oxygen, which has a different vibrational value and appears in a different place. And we can use reference tables to identify where particular functional groups should appear on the spectrum. So here are some examples. Here's propanol with an OH group, and this is the OH stretch, this peak here. Here's a very similar molecule. We've got 3 carbons again, but now we have a carbonyl group and its peak appears here. Cyclohexene, we've now got no oxygens. We have a double bond. It appears here, near the middle of the spectrum, and the CH stretches cause these peaks here. So here are some pharmaceutical molecules. They're all quite complex compared to the simple things that we've been looking at. And actually, they have similar functional groups. But we can look in detail at the molecule and we can start to see how these functional groups are appearing in the spectrum. So here we've got a carbonyl group, and we see a single carbonyl peak in the middle of the spectrum. In aspirin over here, we've now got two carbonyl groups. And so we see two peaks in that region. Paracetamol, we just have one. It's appearing here in the middle, and we have lots of others arising from the CH bonds within the molecule. If you look at the fingerprint regions, the exact pattern of peaks is different for each molecule. And the overall pattern is different for each of these molecules, which have similar functional groups. They all have a benzene ring, they all have a carbonyl group, and they all have an OH group of some type as well. It would be very difficult using chemical tests to identify which of these molecules was present in our sample. But using infrared, we have very distinct patterns. So it's quite easy using infrared to identify which sample we have. So this is really important in pharmaceutical testing. And we use infrared in lots of ways in pharmaceutical science. So we can use it to check if we're in a manufacturing plant, if we're buying in a raw material, or if we've carried out a synthesis reaction to make the molecule that we want, the drug that we want to sell as a medicine. We can check that we have the right material and that the reaction's gone right and we've made the right thing. We can check for impurities. If there were different molecules mixed in the sample, we would start to see different peaks appearing in the trace. We can check for functionality. As a synthesis reaction is happening, then new peaks will be appearing as the molecule is formed. And we can also check for degradation. We store medicines for quite a long time, both in the pharmacy and then in our homes. So we need to know that they're stable. If the molecule starts to break down, then peaks will disappear. If a molecule gets oxidized, for example, new peaks will start to show in the spectrum. So we can check for quality of our medicines over time. We can characterize samples, not just of the raw material of the drug powder, but also tablets and creams, things that we're using. We can check for degradation and we can check what it is. So this is used by the police, for instance. If they arrest somebody who's dealing in drugs, they can see what's in the tablets that are being sold using these sorts of methods. And there are lots of other materials that we use around pharmaceuticals. We very often have them in plastic containers. Tablets are in blister packs. And these can also degrade or react with the drug material. So we can check the packaging and the coatings which are on medicines. So that just gives you an overview of how infrared spectroscopy is used in pharmaceutical science and lots of other sciences as well. And you can follow Absolute Chemistry on Instagram and on Twitter, and you'll see the things that we're doing with schools if you do that.
Thank you both for your insight on the course, Linda and Ryder, for your information on the course modules as well. It's really important and informative to find out those particular modules that you can study and how that ranges from GCSE to A-level up to university level. And I'm sure there'll be some people who are really interested in that. We hope you find today's webinar useful. As I mentioned before, if you are watching on demand, you can ask questions to outreach@ljmu.ac.uk, or you can contact scienceadmissions@ljmu.ac.uk. We do have further talks coming up on the Lightning Lunch series, and you can register for those on our Outreach virtual on-demand page, or you can check them out once they're published on our YouTube account. But from me and from everyone here, thank you very much for watching, and we hope you have a lovely rest of your day.